Okay, so that's it for technologies on transcriptomics. Let's now look at proteomics, which is, of course, the study of all of the proteins in a system. It can also start to look at their structure and function. That's kind of a little other subsection where we start getting more detailed in not only what proteins are there, but how they work. So again, the proteome, the whole set of proteins, is all the proteins that's produced, or if they are modified by an organism, or again by just a system. It confirms the presence of the protein, and it also provides measure or quantity of the protein that is present. So that's another nice thing about proteomics, is it's not just giving you a yes or no if it was there, it's giving you how much protein is there. Just like transcriptomics, similarly, you can collect these from any organism, tissue, or a fluid like urine or saliva or your blood. Again, it's going to be easier to focus when you're looking at a large organism on things like just the proteins in a particular tissue, just the proteins that are in the blood, etc. Whereas if you look at just a cell or just a small organism, you could collect the proteins from the whole organism and look at what's happening. Like transcriptomics, proteomics is also dynamic, right? It gives you a snapshot into what proteins are being expressed right at that moment when you are collecting the sample. So because of this, it's more complicated than something like genomics, because again, the genome is constant, whereas the proteome is going to differ from cell to cell and from time to time, right? Again, as I said before with the transcriptomics, your liver cell is going to be utilizing different enzymes and different proteins than your heart cell is. And not only will different tissues use different proteins, but tissues will use different amounts of a protein at a different time. For example, your pancreas might upregulate or downregulate your production of insulin based on how much sugar is around. So we know that the pancreas will make insulin, but depending on the time that you sample cells from a pancreas, the insulin levels might be higher or lower. So again, it's a snapshot at that time, and it's very dynamic. So similar to transcriptomics, one of the ways that we use proteomics is in comparison type of studies. Usually it's going to help us identify main proteins in a particular sample, and proteins that are expressed differently in related samples. So again, an example here would be disease versus healthy tissue, how those proteins are being differently regulated. A lot of studies will, for example, look at normal cells versus cancer cells. Remember again that these cells have the same genetic code. They have the same blueprints of DNA, but the amount of proteins that they're making, what they're expressing, what they're doing is very different from each other. So something had to happen to make them different, and we want to kind of look at what is different about them first to figure out what is going on. Proteins that are found in, for example, diseased samples can be very good because they can act as a drug target or as a diagnostic marker. So for example, here we're looking at a normal cell versus a cancer cell and a particular protein called the Her2 protein. Both of these cells make the Her2 protein. They have the genetic code to make it, and they both make it. But in cancer cells, it was found that this protein is amplified, meaning there is much more of it. And you could figure this out in a proteomics study, right? You could say, here's my healthy tissue, here's my cancer tissue. Hey, this Her2 protein is way more elevated in cancer cell. This can act as a biomarker. So if we were to collect information from a patient and they had elevated Her2 protein, we would know then that it's potentially related to this type of cancer. Also, we can use these as drug targets. Maybe this particular protein helps a disease function or helps the disease do what it does. So you can target that protein that is special or upregulated in a disease to take down the particular disease. We also know that while profiling proteins, we could potentially figure out functional relation. So proteins that are expressed similarly at similar levels might be functionally related to each other. So you could, instead of figuring out a biomarker for disease, you could figure out how a particular mechanism of protein works. You could say, hey, I noticed that when I eat glucose, both of these proteins get elevated. So they must be both related functionally to glucose. Okay, so generally proteins can be detected using, I'm going to say, general antibody technology. So there's a lot of antibody technologies. These can sometimes be called immunoassays or antibody arrays. They act very similar to that microarray I showed you, where they're going to be having a whole set with each well has a different antibody. Every antibody is good for one protein, and it will tell you whether or not that protein is there based on if the well turns color, if it attaches. The other main technology that we use is mass spectrometry, which some of you guys said. Now, I do want to clarify here, because some of you guys said this isn't a detection technique that's going to be good for like detecting specific proteins. But when you have a complex sample, you do usually need to separate it out first before we analyze it, especially for mass spec. So we will separate all of the proteins away from each other and then do analysis to see what protein they are using mass spectrometry. And one of the ways that we do this, the main way we do this, is by doing a gel. Sometimes it will be a one-dimensional or a two-dimensional gel to help separate the proteins. Now, again, if you guys look at this, this is a two-dimensional gel that you're seeing here. It's very hard to tell exactly. Like, I couldn't take this gel and say, that protein is IHC1 protein, or etc. It's very, very, very complicated on a gel that has so many proteins to tell what every single protein is. So I want to remind you guys that some of these technologies that are really good when you just want to look for one protein or maybe just a couple proteins don't work very well when you're literally looking at hundreds or thousands of proteins, because it's very hard to separate them out. So you need to have a technology that's very specific and that you can do on a large scale. So again, this is something like an antibody array would look like. Here you can see these are two different antibody arrays. One of them is using these A431 cells. So these are the normal cells. And then these are the cells that have been amplified in some way. In this case, one of their proteins have been altered. So they're looking at how the proteins respond when you look at the normal cells versus this situation of an amplification. Similarly, down here, same thing, we have our normal cells. These are called HT29 cells. All of the proteins that they're expressing on this antibody chip. And then what happens when we give them UV light. So these are cells that have been exposed to UV light, same cells, but just exposed to UV light. And we're going to compare. You can see, for example, these two proteins got elevated. So this is actually a pretty small antibody chip array. You can have them larger. Remember, if you really want to look at hundreds of proteins and what's happening with them, you'd have to have a very big chip. So one of the limitations with the antibody chips is again, just like with the microarray chips, you have to have a huge chip that literally has, you have to have an antibody for every single protein on that chip. So they can get very costly, and they can just be very hard to make because you need to make sure you know every single antibody that goes to its corresponding protein. So if you have a protein that, for example, we didn't already know about, if there is a protein that's new, a protein that's specific to only cancer cells that doesn't get made in normal cells, then your antibody chip might not pick it up. It probably won't pick it up because we didn't already know to have an antibody for that new protein that nobody knew about. So the best type of technique in this case would be mass spectrometry. It's not as accurate. So antibody tests are very accurate. They will definitely bind the protein. But mass spec is going to let you look at all of the proteins. It's going to give you all the information. So it's similar to when we do DNA. It's like doing the whole sequencing of the DNA, or the RNA. So like RNA-seq. In these types of experiments, again, you just are extracting your proteins. There are two types of mass spectrometry, two types of analysis you can do. They're called bottom up and top down. I won't get into exactly what you do there, but generally just know there's the two different types, and they are going to take some type of spectra and then give you all of the proteins that were in a sample from that spectra.